The last lecture, the last video was on the humoral immune system. This is going to be the cellular immune system. The cellular immune system, again, focuses on fighting pathogens that are hiding inside your human cells. And the main players are going to be T-cells. So there's a number of different styles of T-cells in your body. We're really only going to learn about two of them. So these two types of T-cells have really essential roles in cellular adaptive immunity. We're going to start with the CD8 plus T-cells. CD stands for cluster of differentiation. So this is cluster of differentiation number 8. The clusters of differentiation are just molecules on the surface of the T-cells, and we've just numbered them. So every T-cell has a cluster of differentiation, and so we've just numbered them, and that indicates which T-cell is which type, and they do different things. So this is cluster of differentiation number 8. Okay. Another name for these kinds of T-cells are cytotoxic lymphocytes. Okay, so the pathway by which these cytotoxic lymphocytes work are this. So your host cell gets infected by some microbe, usually a virus, but some bacteria can also enter your host cells. The host cell, finding itself infected, displays an antigen from the pathogen that's infecting it on its MHC class I molecules. So if you remember from the last video, MHC class 1, or from the vocabulary video, MHC class 1, that stands for Major Histocompatibility Complex class 1. And remember, the MHC class 1s are the self-tag. So usually they indicate to your immune system, I belong here. But in the case of internal infection, all of your cells can present signs of infection by displaying antigens on the cell surface in the MHC class 1s. So the host cell gets infected by a microbe. That host cell displays an antigen from the internal infection on the MHC class 1s. A CD8 plus T cell, also known as the cytotoxic lymphocyte, or CTL, is going to bind the antigen presenting MHC class 1. So what the cytotoxic lymphocytes do is they just go from one of your normal cells to another, checking the MHC class 1s. They check this one, oh, there's no antigen there, it must be fine. Check this next one, nothing there, it must be fine. Check this next one, oh, there's an antigen there, this cell must be infected. It must have a pathogen hiding inside of it. Okay, so the host cell gets infected, host cell displays antigens from the internal infection on MHC class 1s. The cytotoxic lymphocyte, or the CD8 plus T cells, are going to bind to the MHC class 1 that has an antigen in it. And that's going to stimulate this T cell, the CD8 plus T cell, to attack the infected host cell using perforin. Perforin is a protein complex that punches holes in a cell membrane. So what that cytotoxic lymphocyte is doing is it's punching a hole in your cell, the compromised host cell. Then it excretes what we call granzymes into the pore that it just made, into that hole it just made. Granzymes, you could also call them proteases. So a protease is an enzyme that breaks down protein. So what this T cell has just done to your host cell is it's punched a hole in it, and then it's spilled proteases into the infected cell, and the proteases are going to digest all the proteins inside that cell. Well, that's going to result in apoptosis, so that cell is then going to die. So what the cytotoxic lymphocytes do is they kill your own cells, but your cells that are compromised. There's something wrong with them. There's a sign of an internal infection. So your cell, when one of your cells is infected, it's an act of self-sacrifice to flag down a cytotoxic lymphocyte using the MHC class 1s, because every time it uses the MHC class 1 to indicate to the immune system that it's been infected, it's always going to die. The cytotoxic lymphocytes are going to come and kill your host cell to get rid of, and that's to contain the infection. So once the host cell is destroyed, well, those proteases might destroy the pathogen by themselves, but if they don't, that host cell is now destroyed, and everything that's inside of it is going to spill out into your lymphatic system, your bloodstream, your body fluids, and then your humoral immune system and B cells can come take care of it. So here's a diagram of this happening. So here you have the CD8 plus T cell. And this, it says it's a dendritic cell, it's an antigen presenting cell, but it doesn't really matter what this cell is. It could be any cell in your body because it doesn't have to be another immune cell. This is a cell that's been infected. So it could be any cell in your body. So don't pay attention to the fact that it says it's another immune cell. So this cell in green is one of your normal human cells, but it's been infected. So what it's doing is it has the MHC class 1s on its surface, and it's presenting this little peptide here. That's the antigen. This is a little piece of molecule from one of the microbes that's infecting the inside of it. Now you'll notice on this in pink, this is the cytotoxic lymphocyte. And it has these two receptors here. One we've already talked about, this TCR, that's the T-cell receptor. So we talked in the first video about T-cell receptors and B-cell receptors and how they have this amazing specificity to bind to a single antigen from a single species of pathogen. So that's what this T-cell receptor is for. It's to bind this antigen and recognize this antigen from the pathogen that's infecting the cell. Right next to that, just adjacent, is the cluster of differentiation number 8. So this receptor here is for docking with the MHC class 1. So these two work together. The CD8 docks with the MHC class 1s. The T-cell receptor looks for an antigen. So what these cytotoxic lymphocytes do is their CD8s bind with the MHC class 1s on every cell of your body, checking them. If the T-cell, so the CD8 binds the MHC class 1 on every cell, and then the T-cell receptor looks to see if an antigen it can bind to is there. If not, this cytotoxic lymphocyte just moves on, looking for infection elsewhere. If there is an antigen here that this T-cell receptor can bind to, then the rest of the process continues, and this cytotoxic lymphocyte is going to destroy this host cell in order to contain the infection. And then here's another diagram of it. So here, you don't have another immune cell, you just have this is some kind of epithelial cell. So epithelial cells are just surface cells. So it could be, the surface of an organ or the surface of a blood vessel. So these are some epithelial cell from some part of your body. And there's this internal pathogen in these red dots are a pathogen, maybe a virus, maybe a bacteria. So all of these cells are infected. Here you have in purple the cytotoxic lymphocyte, the CD8 plus T-cell. And this little receptor here is docking with the MHC class 1s. This receptor here is the T-cell receptor that can bind with a very specific antigen from a very specific pathogen. And in the MHC class 1, that's in yellow. In the MHC class 1, you have a little red antigen from the pathogen that's inside. So the CD8 plus T cell found an antigen in this MHC class 1. So it's destroying the cell. So here it's punched a hole in it using perforin. So perforin sounds like perforate. When you punch a bunch of holes in something, you're perforating it. So perforin is a protein that perforates the host cell, and then it's going to spill granzymes into it, which are going to digest all the proteins inside that host cell. Once it's done that, it's going to move on and check here, and it's going to find another infected cell here, so it's going to do the same to this next infected cell. Then it moves on. So all of these cells have been infected. If they're infected by a virus, the virus has spread out to all of the neighboring cells in this area, so the cytotoxic lymphocyte has to go along and kill all of them. And we have one more image of this here. So there's two routes for cytotoxic T cells to work. I'm just working with the route B in this slide. So down here in the route that's circled in red, you have, again, this is another immune cell that's been infected. It's using its MHC class 1 to bind with the T cell receptor here, but here you don't see the cluster of differentiation, do you? But it would be there too to help the T cell receptor dock. So that cytotoxic lymphocyte is going to check here. There's an infection, so there's going to be an antigen presented in that MHC class 1, and so the cytotoxic lymphocyte is going to spill its proteases and granzymes into this infected cell and kill it. So in these slides, you'll notice that sometimes the infected cell is just a normal everyday cell of your body, and sometimes it's another immune cell. There are pathogens that specialize in infecting your immune cells, like HIV, for example, specifically targets immune cells. So just because it's an immune cell doesn't mean that it's phagocytized this pathogen. The MHC class 1s are used to talk to cytotoxic lymphocytes when the host cell is not in control of the pathogen. So we talked about MHC class 2s in the humoral immune system. So remember that white blood cells called antigen-presenting cells, they phagocytize a pathogen, they digest it, and then they talk to helper T cells using their MHC class 2s, right? When they use an MHC class 2 to talk to a helper T cells aren't going to kill them. Helper T cells help amplify the immune response to that pathogen. Because an antigen-presenting cell that's phagocytized and destroyed a pathogen, it's working. It doesn't need to be destroyed. But if that antigen-presenting cell has not phagocytized a pathogen but been infected by a pathogen, that means it's not in control of the pathogen, it's losing the war, and it's been compromised. Then it needs to use the MHC class 1s to talk to the cytotoxic lymphocyte, which is going to come and destroy it because it's been compromised. So if the immune cell is in control of the pathogen, it uses MHC class 2s to talk to helper T cells, which will help them to amplify the immune response. If they're not in control of the pathogen, if the pathogen is infecting them, then they use their MHC class 1 so that the cytotoxic lymphocytes can come and destroy them. So MHC class 1s, every cell in your body has them. Immune cells have them, epithelial cells have them, every cell in your body has them. MHC class 2s are only the antigen-presenting cells have them. So right now for the cytotoxic lymphocytes, they only communicate with the MHC class 1s, the self tag, which any cell in your body can use to flag the cytotoxic lymphocyte, say, I've been compromised, I am infected, you need to come and destroy me before this infection spreads. So I've mentioned apoptosis a couple times, so I want to talk about what exactly that is. I'm sure you've heard it before. Apoptosis is programmed cell death. Some people call it programmed cell suicide. So what happens during apoptosis is the cell's genome gets cut into fragments, the external membranes start to bulge, we call it blebbing. There are signals displayed that attract phagocytes. So as the cell succumbs to apoptosis, it releases signals that will attract phagocytes that will come eat up the cell fragments and digest them and recycle that material, but also digest any pathogens that might have been inside that cell. So the phagocytes digest the remains prior to leakage. And that, again, that's to contain infection. It can be to recycle materials, but it can also be to contain the infection. So healthy cells, apoptosis isn't something unique to infected cells. Healthy cells are also programmed to die at certain intervals. If your cells were not programmed to die, you could not control growth rate of different organs. And that means you couldn't maintain healthy cell numbers and the proper ratios of different cell types between organs. So you can sort of think of this concept as like sculpting. So when you're sculpting something out of clay, sometimes you need to add clay, but also sometimes you need to shave it away to get the proper shape that you want. And in the same way that you might have to shave away clay, you might also need to have apoptosis to help control the form of your body and your different body organs. But apoptosis it can be programmed cell death just to help maintain healthy tissue balances, but it also is stimulated when there's something wrong with the cell, if everything's working properly. The second time, now I want to be clear that there are many kinds of T cells. We are learning two, because the immune system is very complex. These are two of the most essential pathways in the cellular immune system, though. So CD4 plus T cells, that's the second kind of T cell I want you to know. So these have a cluster of differentiation that has been numbered number 4. Another word for these T cells are the helper T cells, and we've actually already seen these. We saw the helper T cells when we talked about the humoral immune response. Remember when B cells phagocytize a pathogen, they then display fragments of that pathogen that they digested on their MHC class 2s. Then they wait for a co-stimulatory signal from a helper T cell, and that helps activate the B cell. So these are the same cells. We've already talked about them in the humoral immunity video, but we're going to tell the story again from the helper T cells perspective. So in this pathway, an antigen-presenting cell, which the only antigen-presenting cell we've covered so far is B cell, but there are other antigen-presenting cells in the immune system that belong to the innate immune system, so we'll get to them after exam 3. So some antigen-presenting cell, like a B cell, phagocytizes a pathogen. The antigen-presenting cell will then present antigens from the microbe it just digested on its MHC class II molecules. Then a CD4 plus T cell, also known as a helper T cell, will bind to the antigen plus the MHC class II. So it's a complex, antigen MHC class II complex. That's going to allow the helper T cell to recognize that the antigen presenting cell has encountered an invader. So you need a co-stimulatory signal from the antigen presenting cell. So the antigen presenting cell, like the B cell, and the helper T cell will stimulate each other using cytokines. So you get this co-stimulatory action. And that's going to cause the helper T cell to proliferate and to secrete cytokines. So when the helper T cell proliferates, some will become various types of T cells, each with different cytokine signals. And some will become long-lived memory cells in your body. So an important part of this is when a helper T cell proliferates, not all of its daughter cells are helper T cells. It can produce other kinds of T cells, but they're all going to have the exact same T cell receptor that allowed it to bind to this MHC class II antigen complex. So just to recap, you have an antigen presenting cell, which the only one we've covered so far is a B cell. So you have an antigen presenting cell like a B cell phagocytizing a pathogen. The antigen presenting cell digests the pathogen and it presents antigens or fragments of that pathogen on its MHC class II. The helper T cell binds to the MHC class II with the antigen in it. Because there's an antigen there, it recognizes that that antigen presenting cell has encountered an invader, so it's going to become activated. So the two cells are going to activate each other. The helper T cell and the antigen presenting cell are going to activate each other. When the helper T cell is activated, it's going to proliferate rapidly and secrete cytokines. The cytokines will help attract other immune cells. Its daughter cells will be many different kinds of T cells, each with different cytokine signals. So different cytokines stimulate different aspects of the immune system. And some of those daughter cells will become those long-lived memory cells that will remain in the body for many, many years, maybe your whole lifetime. An army of T cells devoted to fighting that very specific pathogen that was encountered in the MHC class II complex, the antigen MHC class II complex. So when we talked about this in the humoral immune system, we were following a B cell, which is one kind of antigen presenting cell. And we said when the helper T cell stimulated it, it also proliferated, right? So when the B cell, so if you branch off into the humoral immune system here, so we're following the helper T cell in this slide, but I want to remind you that the antigen presenting cell, when it's a B cell, it also proliferates rapidly. Some of its daughter cells become antibody producing factories and they spit out tons of antibodies into the body fluids, and the other daughter cells become long-lived memory B cells, which are specific to that one species of pathogen. Okay. So in this pathway, not only have you created a whole bunch of long-lived B cells that are devoted to fighting that pathogen that will live in you for years and years, maybe your whole lifetime, but you're also developing an army of T cells, helper T cells, which are devoted to that pathogen. So those are the only two types of T cells I want you to know. I'm going to introduce one more cell type called a natural killer cell. And different textbooks will include these in either the innate immune system or the adaptive immune system. So they're kind of it's unclear where to classify them. And the reason is, if you look at their cell heritage, if you look at what stem cells they come from T cells. And they work like T cells in many ways. But so that would place them in the adaptive immune system where T cells are. But some people would classify them in the innate immune system because of how they behave. Natural killer cells are not specialized to individual pathogens. They have a generalized response to all pathogens. So they always do the exact same thing. B cells and T cells, remember they have those unique T cell receptors and B cell receptors that are specific to one species of pathogen. Natural killer cells don't do that. They can't differentiate between different kinds of pathogens at all. So based on their cell heritage, they're more closely related to T cells, but based on how they function, they act more like an aspect of the innate immune system. So I'm going to introduce them here, and you'll see them again when we get to the innate immune system. So what a natural killer cell does is it distinguishes normal cells from infected cells or abnormal cells. And they do this by looking at the MHC class 1s as well. So remember, cytotoxic lymphocytes also communicated with the MHC class 1s. These are the self tags, right? These are the name tags that say, I belong here. When a cytotoxic lymphocyte docks with an MHC class 1, it looks for an antigen from a pathogen being presented there. That's not what the natural killer cell does. A natural killer cell just looks to see if the MHC class 1 is there or not. So if there's no MHC class 1, the natural killer cell believes you don't belong here. You don't have the self tag. You don't have the name tag that says I belong here. Okay? So if the MHC class 1 molecule is absent, if the cell it's checking doesn't have an MHC class 1, then it's going to kill that cell in a manner very similar to the cytotoxic lymphocytes. It's going to perforate the cell with perforin, and it's going to spill proteases into it to digest all the proteins on the inside of it. If the MHC class molecule is not absent, if the MHC class 1 molecule is there, then it says, oh, you have the self tag, you belong there, and it leaves the cell alone. So the targets for natural killer cells would be things like eukaryotic parasites. So we've been most of the time in this class we're talking about bacteria. We've talked about viruses a little bit. Those bacteria and viruses are much too tiny for natural killer cells to do anything with. But when you're talking about a fungus or a protist, those are eukaryotic parasites, or even a parasitic worm. Those cells and those organisms are much, much bigger. They're much too big for a phagocyte to eat because they're as big as the phagocyte or bigger. So a phagocyte can't go ingest and digest a eukaryotic parasite. So that's what a natural killer cell is for. So a natural killer cell will go up to those large fungal cells or protist cells or parasitic worm cells, check for the MHC class 1 that they're familiar with, and when they don't find it, they're going to attack those cells. They're going to punch holes in them and fill them with proteases. Another target for natural killer cells are tumor cells. Sometimes, not always, but sometimes. So you know that tumor cells are created from over-replication of your own cells, your own normal cells. Something's wrong with the genes that regulate the cell cycle, and they start proliferating and dividing much too rapidly. Well, a lot of times the problems that tumor cells have, they accumulate a lot of mutations, and that can make them produce weird things, look weird ways, and often tumor cells are lacking MHC class 1s. They don't have as many MHC class 1s as a normal cell is supposed to have. So if a tumor cell is lacking MHC class 1s or it doesn't have enough MHC class 1s, natural killer cells will kill them. So they can help fight tumor cells as well. And the final target for natural killer cells are viral infected cells. So now you're going to learn a little bit about this back and forth of the arms race. So I told you that you have a humoral immune system that attacks pathogens in the body fluids. But then you have pathogens like viruses that hide from the humoral immune system and hide out inside your normal cells where the humoral immune system can't reach them. So then you have a cellular immune system that the normal host, the cell that's infected, the host cell that's infected can flag down the cellular immune system by presenting antigens on the MHC class 1s. Cytotoxic lymphocyte comes, kills the infected host cell. Well, some viruses have evolved the ability to knock out the host cell's ability to even have an MHC class 1. So it says you're not going to show the cytotoxic lymphocytes that you're infected because I'm going to take away your MHC class 1s. Now you can't talk to cytotoxic lymphocytes anymore. Well, then now you have natural killer cells that are checking for, to make sure that all the cells in the body have the appropriate self tag. So a natural killer cell is going to come and say, the MHC class 1 is missing. That's highly suspicious. I'm going to kill this cell. So a virus can try to hide from the cytotoxic lymphocytes by knocking out the MHC class 1s, the antigen display case, but then the natural killer cell will come and say, this is missing the self tag, so it'll kill the cell anyway. So a brief review of everything we've covered for the adaptive immune system. Remember, you have two immune systems. You have your innate immune system, which we'll cover after exam 3, and you have the adaptive immune system, which is what we're covering this week. The innate immune system is an immediate but a non-specific response to infection, and it's actually your first lines of defense. So pathogens encounter your innate immune system first. The adaptive immune system is very, very slow to gear up, but it's a highly specific response to infection, and it involves memory of previously encountered pathogens. So you generate this, when the adaptive immune system comes into play, it generates this chemical memory that allows the immune system to recognize a pathogen the next time it encounters it, and the second encounter, it will remember it so quickly and be able to fight it so quickly that you probably won't even notice that you've been infected by the pathogen the second time you encounter it, because your adaptive immune system, upon the second encounter, can nip the infection in the bud, because it generates those armies of long-lived memory cells, B cells and T cells, to fight that pathogen. So there are two branches of the adaptive immune system. There's the humoral and the cellular. In the humoral immune system, what the humoral immune system does is it attacks pathogens that are outside the host cells, that are floating around in your body fluids. The main players are the B cells, but the helper T cells are secondary players that assist. And B cells produce immunoglobulins. Immunoglobulins can be B cell receptors or antibodies. And remember, the receptors for pathogens, for antigens on the immunoglobulins are incredibly diverse. So for the B cell receptors, what they do is, so when a B cell receptor binds with, the B cell receptor is an immunoglobulin embedded in the B cell surface. And when it binds to an antigen, it's going to induce phagocytosis of the antigen with the pathogen attached. The B cell is then going to digest that pathogen. It's going to display antigens from the pathogen on its MHC class IIs. The helper T cells then are going to come bind with the MHC class IIs, find the antigen there, and that's going to cause them to release cytokines, which stimulates the B cell to proliferate. It's going to proliferate a whole bunch of perfect clones that have the exact same B cell receptors that helps them bind to and phagocytize the pathogen. Some of the daughter cells will be memory cells that live in your body for years, and some of them are going to be antibody-producing cells, which are their antibody factories. When an immunoglobulin is released into the bloodstream or into the body fluids, it's called an antibody. But it's the same thing as a B cell receptor, it's just let loose. There are 5 things that free-floating antibodies can do to fight infection. Agglutination, which is causing the pathogens to all clump up together. Opsonization, where they act as grips to facilitate phagocytosis of the pathogen. Antibody-dependent cell-mediated cytotoxicity, that's where they stimulate an immune cell that can produce antibiotics to spill out its toxic antibiotics onto a pathogen. Neutralization, that's when the antibody binds to and neutralizes an exotoxin, or binds to and neutralizes adhesins that help the pathogen adhere to your cells. Or activate the complement system. That's just activating a branch of the immune system that we'll cover later. The cellular branch of the adaptive immune system primarily attacks pathogens that are hiding out inside your host cells, but it can also assist with humoral immunity. So the main players are T cells, and T cells have T cell receptors, which are also incredibly diverse in terms of the binding sites to pathogens, which make them highly specialized for individual pathogens. And we have covered 2 types of T cells, although there are many more. We've covered the CD4 plus T cells, which are the helper T cells. The helper T cells assist with the humoral immune system. So what they do is they recognize, and they'll also assist, you'll find later, they will also assist with the innate immune system. So they basically help out with all the different branches of the immune system. That's why they're called helper T cells. So those are the CD4 plus T cells. So what they do is they recognize a foreign antigen that's presented on class II MHCs on antigen presenting cells like B cells. And then they release a co-stimulatory signal to stimulate the antigen presenting cell, and then they also become stimulated. The helper T cell then proliferates into many types of T cells, and each different kind of T cell produces its own cytokine, which activates its own branch of the immune system. And some of the daughter cells also become those long-lived memory cells that will sit in your body laying in wait in case that pathogen ever dares to come back. And so this process, the whole cytokine releasing process, is going to activate the innate immune system. It's going to activate the B cells, the humoral adaptive immune system. So basically these are just cytokines to activate different branches of the immune system. The CD8 plus T cells are also known as cytotoxic lymphocytes. The job of cytotoxic lymphocytes is to destroy self cells that are corrupt. It recognizes corrupt cells by foreign antigens presented on the class 1 MHCs. When it recognizes that a host cell is corrupt, it's going to perforate the host cell and spill proteases to cause apoptosis of that corrupt cell. In this slide, you can see the difference between the cytotoxic T cell and the helper T cell. So in both cases, they're binding to, so in the cytotoxic T cell diagram, you don't know what kind of cell this is. It could be an immune cell, it could be any cell in your body. So this is a cell that's been infected. Because it's infected, it's not in control of this pathogen. It's using its MHC class 1 to flag down a cytotoxic lymphocyte. Once the cytotoxic lymphocyte binds to an antigen in the MHC class 1, it's going to kill the host cell, and that's going to help contain the infection. It's going to expose, it's either going to destroy the pathogen with the proteases, or it's going to expose the pathogen to the humoral immune system. In this part of the diagram, you see this is specifically an antigen-presenting cell, which is an immune cell, because only antigen-presenting cells have the MHC class 2s. The antigen-presenting cell's job is to phagocytize pathogens. So it ingests them, digests them, takes that digested material, displays it on the MHC class 2s, and it's using MHC class 2 because it is in control of the pathogen. It's already destroyed the pathogen. This cell is working just fine. You do not want a cytotoxic lymphocyte to come and kill it because it's working perfectly. So it's going to display fragments of what it digested on the MHC class 2s. That's going to help it communicate with helper T cells. The helper T cell is going to activate the antigen presenting cell. It's also going to become activated, and it's going to proliferate. So cytotoxic T cells are for when the cell is corrupt, when it's been infected and it's not in control of the pathogen. Helper T cells are for when an antigen presenting cell, an immune cell, is working just fine and it needs help to become activated. Cytotoxic lymphocytes communicate with MHC class 1s, which all cells have. Helper T cells communicate with MHC class 2s, which only antigen presenting cells have. And the last bit of review is for the natural killer cells. The natural killer cells come from a lineage of cells that are part of the adaptive immune system, but they behave more like a part of the innate immune system because they have a generalized response to infection. They're not specific to individual species of pathogens. So it's kind of an innate response. They destroy parasites and also corrupt self-cells, like tumor cells or cells that have been infected by certain kinds of viruses. They recognize problems when cells don't have the self-tag. So they recognize a problem by looking for MHC class 1s. And if a cell does not have the self-tag, does not have the MHC class 1, it's going to destroy the cell. Either it's a corrupt host cell or it's a cell that doesn't even belong to the body. And they kill in a method similar to the cytotoxic lymphocytes. They perforate and they spill proteases into the foreign cell or the corrupt cell.